Hello, everyone, and welcome back. We're going to be talking about endo and exocytosis. Now, remember, we've talked about this before that anytime you see endo, I want you to think about going inside, and exo, think about exit, going outside. Okay? So, some cells have a special type of active transport. So, remember, active transport needs energy. And it's called endocytosis and exocytosis. In endocytosis, cells engulf substances. So engulf means to wrap around, okay, to completely take over. This is to engulf. In exocytosis, cells expel substances or things. So expels means to push out, to get out. Okay, so we're going to talk about three types of endocytosis and one type of exocytosis, okay? So let's see. Endocytosis allows cells to take in large molecules that cannot pass across a cell membrane or through channels in the membrane. So basically, this allows the cell to take things inside that are too big to go in through a channel protein or through osmosis and things like that, okay? There are three major types of endocytosis. So remember, endo means going inside. Phagocytosis, pinocytosis, and receptor-mediated endocytosis, okay? The first one is phagocytosis. Basically, in this process, the cell engulfs a solid particle. So basically, here's the thing right here that it's trying to grab, and the cell will extend out and wrap around it and then take it in. You can think about it like the cell is eating it, okay? For this reason, phagocytosis is called cell eating. So phagocytosis is also used by white blood cells as they take in or engulf diseases or bacteria or things that might make us sick. So white blood cells go through our veins, our bodies, and then they engulf bad things. Like, for example, if this is a disease or a virus, our white blood cell will take it in and destroy it. Okay, so that's phagocytosis. Main thing to think about, it's like cell eating, okay? It'll reach out right here and grab whatever it's trying to get. Pinocytosis is a way that cells bring certain particles into the cell. So the cell membrane invaginates or pinches around the particle. So first, there's a particle and it invaginates. It makes kind of like a bag, all right? And eventually, it pinches until this part here closes and turns kind of like into a little bag. And then the particle goes inside the cell, okay? This process is often used to take in fluid or liquids from outside the cell. For this reason, pinocytosis is also called cell drinking. So remember, in phagocytosis, this right here is reaching out. For pinocytosis, it's a bag that wraps around what the cell's trying to get in. Okay, so if it's trying to pull in some sort of fluid, it would take it in like that, invaginate, and then it would eventually close off and let it go inside the cell. This is the long one. Receptor-mediated endocytosis is a very special type of endocytosis. In receptor-mediated endocytosis, substances, they connect to receptors. Remember, receptors are things that are receiving information or things on the membrane. Once these receptors are full and they have what they need, it makes the cell invaginate. Remember, it turns into a bag. The cell then it takes in the particles into a little bag as it holds onto it with the receptors. Okay. This form of endocytosis is often used for cell communication. Okay, these receptors often they connect to hormones. So there will be a hormone outside the cell, it'll connect, and that hormone will be taken inside the cell, which will affect how the cell grows and behaves. Okay. Exocytosis, so think about exit. Okay, it's a process that expels or gets out things from the cell. Okay, it's an important way to get rid of waste from the cell. It's also important to release proteins made by the cell. This is the most common way cells release hormones or enzymes. So remember, enzymes help chemical reactions happen faster within cells and in your body. Exocytosis occurs in 3 steps. Step number 1, the Golgi bodies package substances to be expelled or taken out from the cell into vesicles. So it packages what it's trying to send out into a vesicle, which is like a little bag. Then the vesicle travels to the cell membrane. The vesicle then connects or fuses with the cell membrane, and then the content on the inside is let out. Okay, so step 1, the Golgi apparatus or Golgi body makes and packages it. It puts it in a little bag that sends it out, and when it touches the cell membrane, it starts to open up and the stuff inside comes out. And that is the end of our talk. That's exocytosis, endocytosis through phagocytosis, pinocytosis, and receptor-mediated endocytosis.